Okay, we're about ready to go here. Oh, I should probably get the thing. Yeah, it's live right now. We'll give it a couple more minutes. It's not quite time yet.
Hi, Professor Foster.
Seems like it. I might do this. Okay, we're gonna go back to muting this. Okay, it is 6, so I think I will begin things out here and begin our introductions and go ahead and get on with the thesis defense tonight. So I just really want to briefly introduce Jaydee, who started off life something like this. To really understand and give a proper introduction, I have to start at the beginning and give you a little bit of a brief introduction to how he got to where he is right now. He started off very small and very cute and in Thailand at this moment. I think, right, in Thailand when this was taken? Okay. And so that's there, if you don't know where Thailand is. From there, he actually went on and did a lot of his early schooling in China. I looked up the specific city, but I will not dare try to pronounce it. From there, he did that until late high school, in which case he went to San Diego, where he finished his high school there, and then after that came up and joined us at Walla Walla University and did his undergraduate here, and then also at the end of his undergraduate, which, by the way, he was majoring in a dual major of bioengineering and computer science, decided that he wanted to do some interesting big data senior project in some sort of biological system. So Dr. Foster, I believe, directed him to me, and in which case we started working on looking at RNA sequencing data. And he decided he really enjoyed that so much he stayed on and stayed with us. One thing I just kind of realized as I was going through and making this map today is that I think this is, despite now working on a marine animal, the furthest you've been away from the ocean. Is that true? Looks okay. So then he started grad school. He joined my lab. We did things kind of like this. This waterfront pizza in Port Townsend, which if you haven't been there, I'd highly suggest it. And I figured out he must be a pretty good guy because my kids really took to him pretty quickly, and they're a pretty good judge of character, I think. Now, as I was going through and putting together this introduction, I went through my phone and tried to figure out, okay, so I want to put together some memories, some things that I have from Jaydee, and I was going through the picture roll from the Facebook Messenger chat. And I found a lot of things I didn't remember exactly what was going on, kind of like this right here. I don't quite remember. I also learned some things from him along the way, like the proper way to eat bananas. But also, it was really interesting because going through, you could really see what we're spending a lot of time on because he would send back and forth pictures about the research he was doing. So there's often quite a few pictures in there that look like this as we are staring at output statistics from various bioinformatic tools as we were going through. He did an awful lot of that, as you're going to see in this thesis. However, that's not all he did. That was a big undertaking in and of itself. But during his time in my lab, he's also done a lot to become a really full-fledged ocean acidification biologist in his own right. It is its own kind of sub-discipline of biology to run ocean acidification experiments, and he's spent a lot of time in the ocean acidification lab running measurements on the treatments. Probably spent way more time than he wanted in places like this right here, which is running titrations on the ocean acidification treatments, which is the bane of many of my students as you sit there monotonously while the ocean is outside. Also, you get an idea of what they're doing with their time by the sheer volume of the kinds of pictures that you get of certain kinds. And I can tell you that he ran a lot of gels. This is actually just a small subsample of all the pictures of gels that I have from him in the course of my picture roll. This doesn't even include all the time that we spent putting into trying to make our own light gel box because the ones that we had going didn't work. And this one actually worked okay, but not all that great as well. So we did a lot of that. But at the end of the day, he did a lot of really interesting work. He was part of probably the most science-y picture I've ever took in my life, I think, which is this. This is him helping out his lab mate, Sophie Sonner, as she was looking at metabolic rate of chromatophores. We had great teamwork in the lab during that time. But at this point, I don't want to take up any more of his time. He's done a lot of work, put a lot of interesting thought into this, and has some great results to share with you. So I'm going to give the time over to Jaydee. Yeah. Go ahead and put that on and give it a couple seconds before just shift stuff over. But go ahead and put it on.
Okay. Wow. Thank you all for being here. I'm not going to miss making those gels. I'll be here in Rigby till like midnight for almost a couple months. Chris knows. Does this work? Okay. Well, thank you all for being here, all the faculty, classmates, friends, and also people watching on YouTube, my family in Thailand, some cousins in Singapore. And today I want to talk about octopuses, and if you haven't already know how interesting and amazing they are, I want to show you that they are one of the most amazing organisms you can find. A lot of people have called octopuses the closest thing to aliens on Earth because of the level of their intelligence and how complex the behaviors are. This is an octopus, astronaut octopus. Okay, this is the slide I wanted to go into. This is an octopus opening a container to get the crabs inside. And not only octopuses can open containers from the outside, they can also open containers or jars from the inside. Here, they can also walk. Is a picture of them walking, but it's actually more impressive than them walking is they're carrying coconut shells, and they use that as mobile shelters. And this is the only example in invertebrates that scientists show that this is the only example of invertebrates using tools. And this is crazy. This is an octopus doing camouflage. They are the best at doing camouflage in nature. And they can just instantly change the texture and color of their skin to match their surroundings. Another amazing thing octopus can do that a lot of people don't know about is RNA editing. So RNA editing, before I go into that, I want to give you some basic molecular biology information because it is a heavy part of my research, and I just want to make sure that you know some of the background before I go into it because I'll just repeatedly bring up those concepts. So first thing is DNA uses 4 bases. It uses adenine, cytosine, guanine, and thymine. RNA also uses 4 bases. They use the same bases, except DNA uses a thymine and RNA uses a uracil. When you have these bases and they pair together, you have A usually pairs with T or U, and C usually pairs with G. And for the purpose of my research, and for the purpose of this presentation, you can think of T and U as the same thing. What you see complementary base pairing is in double-stranded structure of DNA and RNA. And also in how DNA is transcribed to RNA, and I'll talk about that a little bit later. Another concept is what is a codon. A codon is a sequence of 3 bases that translate to a protein. So to put all of that information together, here you have a double-stranded DNA, and the bottom strand of the DNA is used as a template to transcribe to the mRNA. Because transcription uses complementary base pairing, you see that the mRNA is actually almost the same as the top strand of the DNA, except that you have T's in DNA, but you have U's in RNA. And then each 3 bases is translated to an amino acid, and you put all the amino acids together, they form a protein or polypeptide. And this is the central dogma of molecular biology, that information is stored in the DNA, and it transfers from DNA to protein using mRNA as a medium. Okay, now we can talk about RNA editing. So there are 2 categories of RNA editing. One is insertion and deletion editing. This is less common because what I just talked about, how protein is translated, if you add or delete a nucleotide or a base, because each 3 is translated at a time, when you add anything that is not a multiple of 3, you completely mess up the frames and you completely change the protein sequence. So this is less common, but there are examples of very useful insertion and deletion editing. More common is substitution editing, and this is the editing that happens everywhere in animals, in viruses, in plants. One type of substitution editing is C to U editing, and this is facilitated by the enzyme APOBAC. The other type is A to U, A to I. I stands for inosine, and this is mediated by the family of enzyme called adenosine deaminase acting on RNA, or Adar for short. And this is the most common type of RNA editing, what I focus on, research. And inosine is interpreted as guanine by biological processes, by sequencing. So for the purpose of this presentation, you'll see me sometimes referring A to I editing as A to G, but just know that they're the same thing. So back to the central dogma of molecular biology, what RNA editing is, so here you have an mRNA that is transcribed and processed, right? And then you can have an ADAR that comes in and just change an A to an I, but I is a G, so you have the codon changes from AUC to a GUC. And what that does is it actually changes the amino acid from isoleucine to valine. So this is an example of have a pullback come in and changes C to U and changes the codon UCG to UUG, and that changes the protein from serine to a leucine. So there are a couple things to keep in mind about RNA editing before I talk about how that's used in octopus. First is, go back to the recoding and non-recoding slides. Yeah, after this. Okay. So what I just talked about are recoding edits. So these are edits that change the amino acids, and because amino acids are the fundamental building blocks for proteins, sometimes you potentially change the functions of the proteins. But you also have non-recoding edits, and these are edits that don't change the amino acids and the proteins. And the reason is that you have multiple codons translate to the same protein. Here you have 4 codons that translate, they all translate to leucine. You can see that the first 2 letters are A and U, and no matter how you change the 3rd letter, you're always going to end up with a leucine. So this is an example of non-recoding edits. Another thing to know about RNA editing is, what RNA editing is, it comes in different percentages or levels. And what that concept is usually you have your mRNA transcripts, and many of them are transcribed from the same DNA. And what an editing level is what is the percentage of those transcripts that is edited. And like I said, editing is not an all or nothing event. You can have 0% editing, or you can have 100% editing, or anywhere in between. So one of the very interesting place octopuses use RNA editing is in temperature adaptation. An aspect of how temperature changes our physiology is action potentials. Action potentials are also called nerve impulses or spikes, and they are basically electrical signals that neurons communicate to each other. This is how information propagates in the nervous system. And what's responsible for that spike is the ion channels on the surface of neurons. Specifically, the sodium ion channels are more responsible for that rising phase of action potential, and the potassium ion channels are more responsible for the falling phase of the action potentials. How temperature changes this is it actually makes the potassium ion channels work a little bit slower. And what that does is it makes the action potential really long, longer than usual, and it makes the falling phase of the action potential longer. And this is a problem because when you receive signals, when neurons want to transmit signals, you have to fire action potentials repeatedly over and over again. And for one action potential to be fired, another one has to come down. So one needs to come down before they can fire another one. So this is a problem, and this usually slows down signal transmission. And we feel this too in our body, right? In the winter, we just feel slower because our body reacts a little bit slower. And this is what you might expect in octopuses, but that's actually not what happened. So researchers look at the protein structure of the potassium ion channels, and they found that the potassium ion channels look a little bit different in octopuses in cold temperatures. So the proteins are different, but when they look at the DNA, they're virtually the same. So how can you have virtually the same DNA but different proteins? Well, we just talked about how the information is transferred from DNA to RNA to protein. And in this case, it turns out their RNA is edited. So this group of researchers look at octopuses from different areas of the world, from colder temperatures to warmer temperatures. And what they found is that in the potassium ion channels, so here on the x-axis, you have water temperature going from -5 to 45, and on the y-axis, you have editing level at this particular site called I321V. 321 means the 321 amino acid in the octopus potassium ion channels. And what they found is in octopuses living in colder temperatures, especially those living in the Arctic and Antarctic, they have high percentage of RNA editing, somewhere between 70% to 90%. But when they look at octopuses living in the more tropical areas, they have significantly lower level of editings, somewhere between 20% to 40%. And in these octopuses living in colder regions, this high level of editing allows them to adapt to the extreme temperature by making the potassium ion channels work a little bit faster. So the action potentials is not as long as they would usually be. And this is probably the coolest thing, the most interesting, coolest thing about octopus that I feel like people don't know about is that they can just go in and change their RNA and change the protein and how they function. And what I just talked about is just one example of octopuses changing one protein to allow them to do temperature adaptation. It turns out octopuses actually have the most recoding events in any organisms. Even though in humans we find a lot more editing sites, but most of them are not recoding and not adaptive, versus you have about 70,000 in octopuses. So people have looked at how RNA editing responds to temperature in octopuses, but no one really has looked at how ocean acidification affects RNA editing in octopus, and this is the purpose of my study. I want to look at how RNA editing is changed in the gill tissues of octopus rubescens because of elevated CO2 conditions. And let me explain another concept, ocean acidification. What is it? So since the Industrial Revolution, we have seen rising level of carbon dioxide emission, at a steady pace, year over year. Actually, in 2019, we had a record emission level at 37 billion tons. And if you look at the historical data, the only thing that slows down carbon dioxide emissions is recessions. So you see that, in late 2019 to 2020, because we had a recession due to the COVID pandemic, and then in 2008, if you look at historical data, this is what you would see. What that does to the ocean is, 30% of the carbon dioxide in the atmosphere is absorbed by the ocean, and it reacts with water to form carbonic acid, and carbonic acid dissociates into bicarbonate and hydrogen ions. And the increase in hydrogen ions leads to a decrease in pH and making the oceans more acidic. And this is why we picked the gill tissues to be the tissues to look at, because gill tissues is the main site for pH regulation in cephalopods. The octopus I study is Octopus rubescens. They are also called the East Pacific red octopus. We collected them on Whidbey Island, close to our university's marine laboratory, near Bellingham and Anacortes. We put each of them in one of these custom cooler tanks, so you can control the pH, the CO2 of these tanks, and they also have seawater flowing in and out. We set the seawater to match the seawater temperature of where they were collected. And 3 octopuses were put in high CO2 treatments, and 3 octopuses were put in low CO2 treatments. And the PCO2 level for the high CO2 is about 150 micro atmosphere, and for the low CO2 treatment is about 700 microatmosphere. So after 2 weeks, we collected part of the gill tissues, we extracted the RNA and DNA, and we sent them for sequencing. Usually when you do editing site detection, having a genome is really nice, but Octopus rubescens doesn't have a genome, so I used a transcriptome approach with matched gDNA and mRNA from the same individual octopuses, and I'll talk about that in probably more details than you want to hear later. And overall, I analyzed about, over 1 billion sequences. Each sequence, 100 base pair or 150 base pair. So overall, more than 100 billion bases were analyzed. So here we have a bunch of similar RNA sequences. And you might ask the question, I just talked about how you only have U's in RNA, but why are there T's in RNA? So when you do a lot of analysis with RNA is usually single-stranded, so they're not very stable. So before they're sequenced, they're actually converted back to DNA, and then you sequence them. You still maintain the information, still all the ACGT, but it's just in a different form. So, again, for the purpose of this presentation, just treat them the same. And what a transcriptome is basically a profile of all the mRNA that's expressed, and you can talk about that in different scope. You can talk about that, all the transcriptome in an organism, or you can talk about all the transcriptome that's expressed in specific tissues of an organism. So in my case, it's in the gill tissues of octopus. And when you have these similar RNA sequences, you can construct a transcriptome based on what majority consensus is. So here we have an example of 3 A's and 2 G's. So the transcriptome using the majority rule, so it has an A in its transcriptome. And here you have more T's than C's, so it's a T in the transcriptome. And here it is G, so you have a G in the transcriptome. Then I align the gDNA to the transcriptome. And what I was looking for is any difference between the DNA and the transcriptome. And you might ask, so here we have a transcriptome that has a G, but the DNA has a lot of A's. And we just talked about the central dogma of molecular biology, how your RNA is supposed to be almost the same as the DNA, right? Yes, mostly they should be the same. But in this case, one of the reasons is that it's edited. And here we have the DNA in agreement with the transcriptome. They all have A's, but then you see a little difference between the RNA sequences and the transcriptome. So in this case, this is just a minor editing event because just only a small fraction of the RNA are edited. Now in the middle, you have the DNA has T's and has C's, so they're not really consistent. And this is because octopuses are diploid and they have 2 copies of DNA. So when you sequence them, potentially you can get the DNA from 1 copy of the DNA, which might have a T, or you can get a copy of the DNA from, that has a C. So that's why, in this case, we don't really consider this an editing event. So how do you analyze 1 billion sequences? I didn't look at the computer and do what I just explained to you. To do this, you have to use computers. So a lot of what I did was, translating our questions about the data, our methods into computer programming languages and let computer do the analysis. And using that transcriptome with matched gDNA and mRNA approach, I found about 9,000 potential RNA editing sites, specifically A to I editing. And about 2/3 of those are recoding edits. And just a reminder, recoding edits mean the editing changes the amino acids to a different amino acid and potentially changes the function of that protein. So I compared the recoding sites in the gill of rubescens to other species that have been previously reported. So here, my data is the one in the middle, the one that's highlighted in blue. And on its left, you can see that a lot of nervous system tissues in cephalopods actually have way more editing sites, actually orders of magnitudes more. They have, tens of thousands of recoding sites. Versus we found, about 6,000. But when you compare that to human, it's actually double of what human has. Human has about 3,000. And I also ran my pipeline, my code for doing the analysis with the publicly available data for the gill tissues in squid, and I find a similar number of recoding sites in squid. So maybe this is just what's happening in the gill tissues of cephalopods. So previous research have shown that Adar, the enzyme that does A to I RNA editing, has preferences. Specifically, Adar tends to edit at places where there is an G upstream of the editing site. And it likes to edit at places that there's not that many Gs downstream of the editing site. And upstream, downstream just mean one base before the editing site and one base after the editing site. And we want to see if we see this in our data, if we see this preference. So here, I looked at 3 sets of data, and I explained to you my process of identifying editing site. I didn't just find A to I edits. I also found other different mismatches, not just A to I, but C to U and G to A. And then another data set is all the potential A to I edits, and also the A to I edits that are differentially expressed between the high and low CO2 treatment groups. And in the at the bottom, you see 4 nucleotides, and on the x-axis, you see the proportions of what they should be. Could we go back to, this one. Actually, one before. Okay, this is the one I want. Okay, so here you see the dashed lines. Those are the proportion of bases that you would expect if they're completely random. And then we look at this, what is the upstream base for each editing site in these 3 groups. And we want to see that, do we see a pattern that matches what Adar editing preferences? So what I want you to focus on is G, because Adar likes to edit, at places that's overrepresented G downstream and underrepresented G upstream. So here we see that in all the mismatches, you have a certain level of G, and then in potential A to I edits, that proportion is lower. And when you look at A to I edits that have differential editing levels between the high and low CO2, you see that proportion goes even lower. And the same thing with the downstream analysis. When you look at, what's the proportion of G in potential A to I edits, and what is the proportion of G in the differential A to I edits, you see that bias is going greater and greater. So this gives me like confidence to believe that what we found are actually A to I editing sites. So I just gave you a lot of data, a lot of my findings, but they are important, but they're not the most important. Remember, what I'm looking for is A to I editing impacted by elevated CO2 conditions? And what I found is there are 136 A to I editing sites that are different between high and low CO2 treatment. More interestingly, 56 of them are recoding sites, meaning they changes the protein functions. So this is the group, the 56 differential A to I editing site is what I'm really interested in, is what I want to know more about. And in the downstream, upstream analysis I just showed you, this is also the pattern closely matches the Adar editing biases. So we want to pick one to verify, and one gene to verify is the methionine adenosyltransferase subunit 2 gene, or the MAT2 gene, and this gene is involved in ATP synthesis, or fundamental unit of energy in our body. The technique I use is poison primary extension. So what it is it's like a similar, similar to PCR. PCR is something that people use to make copies of DNA. And for these reactions, you all need, the template, the primers, the polymerase, the nucleotides, and the buffers. The difference is for poison primer extension, or PPE, you use a DDNTP as an extension terminator instead of using all 4 DNTPs. Here's an example of an edited transcript. And here I picked the extension terminator to be DDCTP because this edit is an A to I or A to G edit, and G and C, they are complementary to each other. So I'm going to show you why I picked this. So here, you have first the primers is going to attach, is going to bind to the transcript based on complementary base pairing. And you design the primer so that they're complementary so they can bind. And then the polymerase is going to attach to the primers and then add nucleotides to extend that primer. And this is how basically you make copies of DNA. First, it's going to add a T because the next base is an A. And then it's going to add, and then it's going to add an A, another A, and then it's going to add a C. So remember I talked about DDCTP is the extension terminator. So whenever that is incorporated, extension. So that reaction is going to stop there. And here we have an unedited transcript. So at the editing site, it's not edited, it's just A. And you see the same process, but this time, the C is going to be incorporated at a different location. Basically, it's usually just a couple bases down from the editing site. Whatever the next G is, when that C is incorporated, you stop the extension. So that might sound all a little bit too technical, but the key takeaway is when you do a PPE assay, the edited PPE product is going to be of a different length compared to the unedited products. And we can visualize these PPE products using a 15% acrylamide gel. And so this is the gel you saw a lot of picture of. So here we have this one right here, right next to the DNA ladder, we have just the 20 base pair primers. And then in the middle lanes, you have a 50-50 of the 22 base pair primers, the 20 base pair primers. 22 base. So what you can see is even with just 2 base pairs of difference, you can still separate them, right? So when a transcript is edited, if it's 100% edited, you're gonna have 100% of the edited PPE products. If it's unedited, if it's 100% unedited, you're gonna have 100% of the unedited PPE products. But if it's edited, somewhere between 0 to 100%, then the proportions of the PPE products between the edited and the unedited is gonna be similar to the editing level. So basically, if something is partially edited, you will see 2 bands. If not, you see 1. And so I ran PPE assay on the MAT2 gene, and this is what I find. And this is the best pictures I can use because of what Dr. Anton talked about, the light box doesn't really work too well with the DNA that we use. So I'm not sure if you can see it here, but the PPE products is where the arrow should be. And here I'm going to show you a better picture. So hopefully you can see here, right there. So I don't know how if you see it, but so that is where the unedited PPE products are. And because we only see 1 band and we know exactly, what's in the transcriptome. So if it's edited, we would expect to see another band, almost right above it. So to independently verify this, we also sent the MAT2 gene for Sanger sequencing. What you get back from is a chromatogram, and you see the chromatogram is the one above. You see 4 lines of different colors, and each color and each line represent a nucleotide. And you can use some software to analyze, okay, based on the peaks, what is the base composition of ACGT at that specific location. So at the top, you have the transcriptome, and the editing site is the first one. And you can see that at this editing site, it's an A in the transcriptome, but it's G when we get back the Sanger sequencing result. So this is an example of A to G or A to I editing. But if you look at the next page, where you get back transcriptome, it's 100% C, but it's also a C in the transcriptome. So they match pretty closely. And if you look at the rest of that graph, that is pretty much the same. So this is one example of Sanger sequencing. All the other samples, they look just like this. So when you compare the editing levels in the MAT2 gene between the original experiment using RNA sequencing and then the verification experiment using both poison primary extension and Sanger sequencing, we see that in the original experiment, we see an editing level that's kind of different between the high treatments. In the high CO2 treatments, you see somewhere between 25 to 50% editing level, and in the low CO2 treatment, it's somewhere between 50 to 75%. But in our verification step, we see that they're all edited, almost 100%. So to conclude, overall I found about 9,000 putative A to I editing sites. About 2/3 of them are recoding. And I found 136 differential A to I editing sites, and 56 of them are recoding. And this is the group that's really interesting. And we showed that with the downstream and upstream analysis, that the putative A to I editing site show RNA editing bias, and that bias is even greater in the differential A to I editing site. And lastly, I showed that the MAT2 gene is verified to be true RNA edited, but in the verification step, it wasn't shown to be differentially edited. So what can you take from here with this research? We only verified one of the 56. And so I think what's really interesting is to look at, how that group of differentially editing sites affect the physiology of octopus and see if they play a role in how they deal with elevated CO2 conditions. Another direction someone can take from this is I show that in the nervous system tissues of cephalopods, you see a lot of editing, right? Orders of magnitudes more. And you can basically run the same experiments, but just using the nervous tissues. And what's good about this is I already wrote the code and everything, and it's published on GitHub. So you can, extract the RNA and DNA from the nervous system tissues and just plug in that data, and you can get that result back in about a week. And lastly, I just want to take a moment to thank everyone who has helped me with my research. First and foremost, my major professor, Dr. Antang. Thank you for all the guidance and advice throughout this project. I also want to thank Dr. Nestler and Dr. Brothers for being on my committees, helping evaluate my research design and giving me valuable feedbacks. I also want to thank Sophie Soner for helping with tissue collections and RNA extraction. And special thanks to Alan Verdi and Jessica Di Oria from Maine Maritime Academy for helping with octopus dives collections. I want to thank Professor Foster for being here as the observer of the process. And thank you. Now I'll open up to questions. Dr. Lindsay?
Do you know what the GC content of the genome is?
Of the octopus genome? Of the octopus genome, or? Well, you mean transcriptome or genome? I don't know. Why?
My question, and I'll tell you why I asked the question. The question is how do they know, how do they show that non-recoding
Oh, okay.
Expression levels in humans, you get things like cancer without changing the actual amino acid sequence. Just drop the expression, you get cancer. So, so my question then is again, how can you say, or how can whoever you're quoting, because I know this is background, it's not directly research, I know that. How do they say that non-recoding changes are non-adaptive?
So the way they define that is they look at the editing level. So kind of in evolutionary biology, there is this idea that if something is useful, it will happen more, and if it's not useful, it will be eliminated. It will not be favored by natural selection, right? And what they look at was they were looking at, okay, what, in the editing sites, what is the editing levels of those non-synonymous or recoding or non-recoding edits, and if the editing levels of the of the non-recoding doesn't go up, then that's how they say that it's non-adaptive, because it doesn't seem to happen a lot more often. Does that make sense to you? But I didn't know anything about how you can have a non-recoding change but still affects the expression of other things. So that's not something I've read in the paper, but
So what I'm really trying to, so one way to look at this is are they calling them non-adaptive because they're assuming they're non-adaptive because they change the amino acids, because that has been how people have reasoned that in the past? Or is there an actual evidence that they're not?
I haven't really seen people talk about how recoding and non-recoding being adaptive or not from that methodology. Really what they look at is just comparing the editing level and see if there is significantly different. In this paper, they look at C to U edits, not just A to I, but C to U edits in human as well. And what they compare is, okay, what is the editing level of the non-recoding, and what is the editing level of the recoding? And if there is a significant difference, at least that's what I took away from that paper. Yeah.
I have one here from online. Judith Humgort asks, fantastic work, congratulations. I was wondering if you had any sense of what kind of genes are edited, or how those might help with adaptation to ocean acidification.
So we know what kind of genes are edited because there's this process called blasting. So once we find an editing site, we know the sequences, we can blast that sequence to a database, and we know what the functions of those sequences are. And so we know the functions are, but the difficult thing is, this is a really, kind of an interdisciplinary project. Just even within biology, you need to know how to code, you need to know the physiology, and then you need to know all these other things. So, we don't really know, for example, what biological pathway they're involved in. I think, those are really interesting to look at. If I have more time, probably look at that. But in a way, it's kind of nice that we didn't find, for example, like 10,000, right? How do you go about finding, okay, what do they do if I give you 10,000 differential A to I edits? Versus if I just give 56, I can probably just, do some gel, run some analysis. That's at least doable with, yeah. Dr. Nestler.
MAT 2, why did you put so much attention onto MAT 2 as to some other transcript?
Okay, so MAT2 is involved in ATP generation, energy, how energy is generated in organisms, and there's a couple things that go into us selecting what candidates to look at. One is of course their functions. Do I know what MAT2 does? And how that helps with pH regulation? I don't know, but we also look at, what is the editing levels in those samples, and did we get DNA coverage? Did we get RNA coverage? What is the RNA coverage? You know, if you just get one RNA sequence and it's edited, is that 100% edited? So, you know, when we make these decisions to look at, you know, what candidates we pick, you know, we look at a lot of different things. But I would say that this project is more, and, you know, understand a whole lot about the physiology of octopuses. But I think, you know, this hopefully, you know, can give someone a good foundation to look at. Okay, you know, we sequence the RNA and DNA, and this is what we find the differences. Dr. Brothers.
So you talked about how the general has this bias about where it works, right? And you confirmed yes in your data that's where it works. I was curious, in the data that were edited or the data set that you found, do you see any bias in the codons that are being edited or the codons that are being produced as a result of the editing? Or you have patterns towards certain types of amino acids, simpler or certain charges or something?
Yeah, not a very rigorous analysis on that, but I do notice something about within the codon, you know, the 3 bases. I think it's usually, like, I see a lot of editing in like the first base. I'm not sure which one, but one of them is more edited. So that's the extent of my analysis on that, but I think that's something interesting to look at. Is there a reason?
First, second, or third place is what you're saying?
Yeah. So I don't remember which, but I did notice something that it kind of leans heavily to one of the locations.
Online, Joan Red asks, what kinds of changes in the proteome have resulted from RNA editing for temperature changes? Has anyone looked at this?
Well, we know what they are, right? Because we find what the functions of them are by you know, blasting them to a database. But a more broader analysis on that, we haven't. And the way to do that, I probably imagine there's some kind of biological pathway analysis where you can plug in all these genes and see, you know, what pathways they participate in. And I think that'd be really interesting to look at. But in terms of, I mean, we have a list, but we don't really know, you know, what pathways and what proteomes they involve in yet. Professor Foster.
You had the 3 and 3 in the 2 groups with the low and high.
Yeah.
And then you found the 56 differences, or the some number of differences. How does that fit into a statistical, what is the statistically significant difference there?
So the first group of, from the original experiments where we did the RNA sequencing on, we actually had, the numbers are a lot higher than that, and that's how we produced that 56 number. And overall, we look at, you know, 50,000 recoding sites, and these are not just A to I edits, but also other modifications. And it's from that group that we found, okay, these are the sites that are differentially edited, and also that's A to I, and also recoding. Does that answer your question, or?
Well, I guess I assume that you're going to get a randomness in the 2 groups. And how do you separate what you observed from what would be random?
Yeah, so that's I mean, a big part of this project is you know, statistic, right? And that's definitely a problem because you might notice we actually didn't do, you know, some statistical analysis on, for example, the upstream downstream analysis. We just observed overall, okay, there is this, and the way to deal with that is just you know, for that specific edits, you know, the editing between in the MAT2 gene. I think originally we tried to use a t-test to see, okay, is there a difference between the high group and low group, but then because of the low sample size, that also kind of violate the normality of the data. These statistical tests, you need to meet certain requirements. What is the sample size? What is, you need to have, sometimes, 10 times, these sample size needs to be independent. We try our best to do statistical analysis, but like you said, when you have a lot of targets to look at, that can be a problem, right? When you have not that many, that can also be a problem. But we end up using a two-tail that differentially edit. Yeah. So the p-value was, I think, let me see, I can go back. Okay, never mind. The p-value for the methionine gene in the Sanger sequencing is about 0.437, if I remember. Yeah, it's 0.05, and it's a two-tail Mann-Whitney test.
And you tried a few others before settling on that one?
Yes, but also with the t-test, the p-value was also greater than 0.05. Yeah, but that's something I want to get good at, statistics, because sometimes I don't know what I'm doing. Dr. Lindsay.
So your work focused on the existence of the editing. What is known in octopus or closely related species about the deaminases? Or is it just an assumption that they're doing that?
What do we know about them? So so Adar I is mediated by Adar, and
Now, they've actually found that in octopus.
yes, and actually Adar is in humans. There are companies that using Adar to.
So my question isn't, do they exist, and do we know what they do in other species? It's what is known specifically about them in octopus. There's a lot of different deaminases. Some of them changes the nucleotide pools. Some of them do these different kinds of editing. So my question is, what is known about them in octopus?
So there are 3 copies of Adar in octopuses. There is Adar 1, there is Adar 2A, and Adar 2B. And on these Adars, you have the RNA binding site, the Z DNA domain. I don't know what that is. And then they're structurally a little bit different, and some of them are more effective at RNA editing than others. Adar 2A is the most effective. Yes. Yeah, and that's actually why you see a lot of RNA editing in the nervous system tissues. Is they find that the Adar activity is just way more in the nervous system tissues. Actually, in octopus, but also in humans. So when you have some something that disrupt the RNA editing level at particular sites, you see that in humans, for example, you get a lot of neurological diseases.
What do you know? Do they know anything about cytidine deaminases, or do they know about other deaminases that have functions, or they just study that particular one?
I think there are studies on those, but for my research, I didn't focus on them because they are not known to be the most common in animals, but there are, cytidine deaminases. Actually, that's the one, a POBAC is the one that does a cytosine to a uracil edits.
Well, ask because I have a very special interest in that particular one.
Oh, why?
Because I discovered that as a finding partner in drug development transition genes that are involved in that. So it seems to be functioning in that. But it's probably one that's changing nucleotide pools, not one that's editing. It's my guess.
Okay.
Just curious what's known in octopus.
I didn't look at a whole lot of C to U edits, but I think that could also be something to look at because just because it's not the most common in animals doesn't mean they don't exist, right? And you can still, look at that and see, how that changes. Jesse.
When were your samples taken of gill tissue?
The original sample. Oh, okay. So Jesse asked, when was the sample taken, the gill tissue sample taken? So the original sample was taken in 2016. That was way before I started the program here. And then the verification experiment, I did that in the COVID summer. That was 2019. Yeah, 2019.
I guess I should clarify, sorry. So, if you go out and collect a sample from a wild-caught octopus, then you put it in a transitioning period of ocean acidification for, like, say, a week, then you take another sample, you're comparing, right?
Take another sample.
Then you do, like, the test period, the trial period, and then you compare the gill tissue from a second sample with that initial first non-edited version. Were you also doing that with your control, where you're comparing after an octopus sat in a cooler for, say, a week, you're comparing the gill tissue to the original wild caught? Or is it just in the ones that were in the ocean acidification?
Oh, okay. I see what you're asking. So you're asking if I'm just try to summarize this. If I'm looking at the RNA only during treatments, or also, when we caught them.
If you took a sample, right, when you caught the octopus, and you put it in treatment, and then you sample it again at the end, and you compare them to look at RNA editing. If you're doing a control and the octopus just sits in a cooler for a week, I want to know if you observed any editing in the octopus that weren't in any treatment, but they were the control at the end versus at the start of the treatment. Did
Yeah.
you see any, like, side effect editing that wasn't actually related to any ocean acidification, or is there none whatsoever?
Yeah, so we try to, so we have the high and low CO2 group, right? And I call them low is because it's relative, but that's really the control group. And what we try to do is when, in the control group, we try to set the temperature in the tank the same as the temperature of where they're caught. And so that's the control group. And we treat them for 2 weeks. And then after that, we compare, after 2 weeks, we compare the RNA editing in, from the control and the high CO2 group. Professor.
How many times did you take the gill samples for each octopus?
We just cut it, and then we use a little bit of it to extract RNA, but we did it only just once for.
Once at the end of the 3 weeks.
Yeah, 2 weeks. 2 weeks.
Did you compare that to initial gill sample?
No.
It was just compared between the high and the low.
Yeah, just the high and low. Yeah.
And what was your sample size?
Sample size, like on what data?
On how many octopus were in each? How many samples did you take from each?
Oh yeah, so we have 3 in high CO2 and 3 in control. But yeah, I think that's one way to look at sample size. But also, when you're sequencing the RNA, right, then you get, tens of millions of sequences. So I don't know how you would define sample size that way, but yeah. But it's 3 in high and 3 in low.
Did you average all 3 octopus for each group, or was there differences in specific editing per octopus, or was it the same between all 3 in each sample group?
Yeah, so this depends on the. Oh, here. So Jesse asked if I took the average of the 3, or if I just do individual. And yeah, for some statistical analysis, we did 3. We did the average. So for example, when we look at the recoding side, what is the editing level between the groups? That's when we took the average. Yeah.
So you didn't notice any difference between
Oh no, there are. But when what I present to you is, it's only like a small subset, but there's a lot of abnormalities in our data, really. So yeah, so there's definitely differences. Yeah.
Okay, well, it is now after 7, so I think we will go ahead and adjourn this portion for now, and then we will have this committee meet after this for a brief, well, probably not quite so brief, but relaxing, question and answer period with him as well. But with that, let's go ahead and give him a round of applause. And
Thank you.
committee members, can we take about 20 minutes here and then reconvene down in the lounge?
Oh, nice. I definitely try to, yeah, make it easier to understand. Thank you.
So much. Oh.
I know, that was,